Well, good morning, good afternoon, everyone. I'm gonna speak first in English and then I'm gonna do it in Spanish. But thank you very much for joining this workshop. This is the last workshop of the Latin American pollen database series that we did this semester. And then today we are very happy to have Ondrej and Suzette Flantua. Ondrej Mottl heads the Laboratory of Quantitative Ecology at Charles University in Prague. And also he's an assistant professor of plant ecology in the Department of Botany. His main areas of focus include quantitative ecology, paleoecology, and data science, among others. And Suzette is a researcher at the Department of Biological Science, University of Bergen, Norway. She's particularly interested in how environmental factors, climate and geology, and human influence have shaped contemporary biodiversity patterns. They collaborate in different projects, and most of these projects have involved large-scale dataset analysis and advanced analytical methods. And together, they have developed R packages for analyzing fossil pollen datasets, one including our FossilPol and another one, which is our RatePol. So today, we're very happy to have them both. They're going to give us a tutorial on how to use our FossilPol. So we know that this can involve some very heavy kind of quantitative data analysis. So if you have at some point like questions, like either you can raise your hand or you can just write your question on the Q&A section, and then we will try to, do our best to answer everybody's questions. Bueno, buenos días y buenas tardes a todos. Muchas gracias por estar aquí hoy con nosotros. Este es el último taller que tenemos de la serie de charlas y talleres que hemos estado organizando con la base de datos de polen de Latinoamérica. Y en esta ocasión tenemos a Ondrej Mottl y Suzette Flantua. Un poco de introducción de ellos. Ondrej dirige el Laboratorio de Ecología Cuantitativa en la Universidad de Charles, Praga, y también es profesor asistente de Ecología Vegetal en el Departamento de Botánica. Sus principales áreas de enfoque incluyen la ecología cuantitativa, la paleoecología y la ciencia de datos, entre otras. Y por su parte, Suzette Flantua es investigadora del Departamento de Ciencias Biológicas de la Universidad de Bergen, Noruega, y ella está particularmente interesada en entender cómo los factores ambientales como el clima y la geología y la influencia humana han moldeado los patrones actuales de biodiversidad. Ambos han colaborado en una diversidad de proyectos que en muchos casos han involucrado el análisis de grandes bases de datos y métodos analíticos avanzados, y conjuntamente han desarrollado 2 paquetes de R para el análisis de bases de datos de polen fósiles, incluyendo FossilPol, del que vamos a hablar hoy, y RatePol. Y bueno, como decía, en esta ocasión van a presentar un tutorial sobre cómo utilizar FossilPol. Vamos a hacer, bueno, la presentación se va a llevar principalmente a cabo en inglés, pero si ustedes quieren hacer también sus preguntas en español, siéntanse libres de hacerlo, los vamos a apoyar también con la traducción. Pueden también levantar su mano si tienen alguna pregunta, o si quieren también pueden poner escribir sus preguntas en la sección de preguntas y respuestas, y vamos a hacer lo mejor para tratar de resolver todas las preguntas, ¿no? With saying that all, thank you very much, Suzette and Ondrej, for being here. So I'm just gonna let you go on and get started.
Thank you very much for a very nice introduction. We will have together a presentation or a workshop about FossilPol, which is the workshop to process global paleoecological data for vegetation-based macroecological synthesis. I'll be speaking English. Suzette can speak Spanish, so you can ask her questions in Spanish. If you're shy, I will not understand Spanish, I'm sorry. Okay, so in today, we're gonna be kind of 2 sections. I think first, we'll kind of show you a presentation, which is publicly available. You will get the link. You can also access it later. And then in the second half, I will show you a little bit practical things, how to actually use the workflow in specifically in RStudio, which is the mostly common used IDE. Okay. So I like to start my presentation with some big numbers, right? And we already know that 7 out of 8 of global Earth boundaries have already been exceeded, which is very, alarming. We also know that more than 3/4 of all ice-free land has been altered by humans, which is also very alarming. But I think there is still a good question, which is when exactly this happened. Has it already happened, with the after the Ice Age, or with the intensive, spread of agriculture, or during the Industrial Revolution? So, and to answer this kind of question, we can use fossil pollen records specifically to get more insight into the past dynamics of the ecosystems. So, as has been said, my name is Ondrej Mottl. I'm assistant professor at the Charles University and head of the Laboratory of Quantitative Ecology. There are some ways where you can contact me, but mostly if you Google my name, you should find my web pages. And Suzette, you want to chip in?
Yeah, and I'm Suzette. I'm at the University of Bergen, working as a scientific professor. I'm the head of a lab which is called Mountains in Motion, and here, like with Ondra, we have just the interactive links to our profile that either on social or on our work that we've been when doing. So yeah.
Okay, so I will start a little bit more broadly about paleoecology, right? So what is it? I guess lots of people know, but I will again. So we use the fossil pollen as kind of a window to past vegetation patterns. Why this is why it's so strong, right? I will do a quick recap of how we are gathering the data, which we're going to be working today. So the main first is we find a specific depositional environment. Mostly it is a lake. And then we will drill a core. And then we will look at the core and take some samples at mostly regular depth intervals. And some amazing people will then look under the microscope and count and identify these individual pollen grains to pollen types. So if we have then the relative abundances of these pollen types in a specific sample, we can then do some age-depth modeling to estimate the ages of these samples, and we will be able to reconstruct the vegetation history of that specific surrounding of that lake, of the depositional environment. And hopefully this can give us a little bit more insight about what was happening in that ecosystem at that specific period of time. Currently, there is over 6,500 individual cores or fossil pollen records with the Neotoma, or this has been 3 months ago, maybe there's even more right now. And this is only, possible through amazing people, individual people who are doing the drilling, the coring, the counting, and then actually uploading this data to Neotoma. So I would like to thank you all for doing this amazing job, because without that, I will not be able to do any of the work which I'm currently doing. But if you have so much data in it and you want to kind of extract the information, then it becomes a little bit more difficult. So if you want to uncover the precise insights from this paleo data, we need tools which will reduce the bias from individual different collectors and samples, etc., to have good data set compilation, which we refer to compilation, where we took several cores together and we want to use the data from them to do some, I would say, macroecological results or patterns. So if you want to build such dataset compilation, there will be several aspects which are actually affecting what the end product, your compilation in the end. That will be the sourcing. Where are you getting the data? Are you getting it only from Neotoma? Are you also adding some of your private data, which you maybe don't want to or you can't even upload to Neotoma? It will be affected by different deposition environments. Was the core taken from a lake? Was it from a mire? Was it maybe from a riverine, from a river? Or some deep sea cores are also available. Then, of course, chronologies. This has been mentioned, and probably quite a lot of you know, because we need to estimate the ages from the individual samples through something called age-depth modeling, and we will a little bit discuss more of that. And there are various methods and techniques how to do so. And if different cores have been used the different methods, there might be some bias in that. So we need to a little bit reduce the bias in different methodology of chronology estimation of these samples. And of course, if we are taking different cores which were identified by different people. There we need to do some harmonization because different people may be may be able to identify to a certain level. Maybe they have differences in their reference collection, etc. So we need to kind of harmonize or unify the names of the pollen types across several cores. And in the end, some kind of filtering. So we want to apply some kind of criteria to make sure that the quality of our data is upholding to certain standards. We would like to do this for macro scale data analysis, meaning we want to look at multiple cores across regions or even subcontinents or even continents or the world. We also want to do it specific for each project, because if you're depending on your research question, you may be interested in various things, and project specific can affect what kind of compilation you want to create. What's for example, the data quality you require, etc., etc. As I mentioned. So we want to make this in a way that we can control for the quality and also reproducibility, which is a big topic for me nowadays, to make sure that anyone else will be able to create the same dataset compilation as you did for your paper or project. And lucky for you, we have created the workflow called FossilPol. It's a workflow to process paleoecological data of fossil pollen for this vegetation-based macroecological synthesis. So all these steps, which I kind of mentioned, the FossilPol is actually taking care of that for you. So we will, in this presentation now, I will use example from Scandinavia, right? Let's imagine we have a project and we want to see, what were the patterns of vegetation changes in Scandinavia during the last 8,000 years? That could be our question. We chose Scandinavia because at the time when we were building this software, we were actually, both me and Suzette were based in Norway, in Bergen. Suzette is still based in Bergen. Okay, so this is the workflow, all the steps that it is doing. So this is very overwhelming figure. So what we're gonna do, we're gonna be taking it step by step, and I will explain each of these individual steps on this workflow and show you what does it mean and how it works. So let's take everything out and let's go at the beginning, which is here on the left up. This is the input, what we are getting in. So what you need to provide as a user are several things. We will now focus mostly on the config file, which is a configuration file, which is basically you are telling the criteria of your, guided by your research question, how the workflow should behave, right? So it controls the behavior of the process with everything which is gonna be doing, and it's reproducible. This is a file where you store all the information. You will see how it looks later, and you can, in the end, you can share it with someone else and say, look, this is the configuration we choose for our FossilPol workshop to get the compilation we have in the end. Okay, I'm not sure. I will pause at some points for questions. This is all very abstract. I'm not sure if is there any question until now? Maybe, yeah, we'll wait a little bit. We'll see. If not, don't hesitate to ask me if something is not clear or I'm going too fast, etc. So,
sorry, I have a question.
Yes.
Your package is
Yes.
meant only for like big amount of data, or just for, I lost that part. Sorry.
Yeah, sure.
But
so
it's like for macroecological data only, or can be like
I would that's an excellent question. And I think if I would suggest you can use it for any data working with 2 or more cores, right? Because if you are at least having 2 cores, then there is a meaningful way of saying, okay, I need to process them in some way that are comparable. You know what I mean? Because if you're just working with single core, that you can do lots of these steps by yourself, or maybe it's not necessary, but I would say 2 or more. And I can imagine if you have only 10 cores with very small region which you are interested in, that this can be used very technically. You can use it for any amount, of course, but it is up to you, right? I would say, but Yeah, hopefully that answered the question.
Yeah. And I have another one. Sorry.
Go for it.
Can I use it for another kind of biological proxy as microinvertebrates, fossils, or something like that? Or is only for pollen?
Excellent. Again, very good question. So currently it is only created for pollen, and we know it works for pollen. It is built in a way that in the future we potentially may expand it with some other proxies, but currently this
Okay.
functionality is not available, right? But if any of you are interested and you want to be coding a little bit, it shouldn't be that hard to do so, but just right now it is only for fossil pollen.
Okay, so it will be a lot of help for me if you could say in your presentation what things are exclusively for pollen.
Okay. Yeah,
Thank you.
I will try. Okay.
Okay, thank you.
I think there are more questions by Alejandra.
Yeah, I do have a question. So you mentioned that, like you using like the Neotoma pollen database. So what happens if, for example, you have these pollen data sets that haven't been uploaded to Neotoma? Can you sort of like mix them, like download some from Neotoma and then add yours? Is it possible?
Excellent. Thank you very much. That's an excellent segue to our next section. And then will be the first step on the workflow, which is the sourcing, which is actually here in this first section of the workflow. It will source all the primary data. The main source is Neotoma, right? Neotoma paleoecological database. But you can also include your own, as you say, private or files, right? They need to be stored in a specific way in either Excel spreadsheets or CSV files. And in the documentation on the website, there is the template to download. And if you put it there and place them in specific folder, they will be both including and processed exactly the same as the Neotoma data. So it is definitely possible. And we in previous papers we have done so. So we used actually both Neotoma and some, for example, private data from Asia to do so. Okay. Thank you for the question. That was great and very nice segue. So in this first section, what will happen, we will gather all the, I would say, primary sources, which is the Neotoma currently and private if available. And then we will do several steps. One of them will be spatial delimitation. So the first step is we need to tell the workflow in which specific area, geographic area, are we interested. So it could be the world, if you don't change anything. It could be very small region, right? As the previous person asked how big it is. It can be very small, specific region. If there are pollen records, it should be possible. And it's up to you. So in this Scandinavian example, back to the example, as I said in the beginning, I chose hopefully you can see this blue rectangle, which was my area of, I say, oh, I'm interested in Scandinavia. And I set this rectangle, this is my focus area. And I run the workflow and it returned all these blue individual dots, which are all fossil pollen records in Neotoma currently. So we are at the first step where we actually source the data from Neotoma and get some first part of the process, right? There's additional things which we can do at this step. There's spatial elimination. There's also selection of ecological groups, which the workflow will ask you which ecological groups do you want to use. And you can say, for example, that you don't want any aquatics, and the aquatics will be automatically filtered out. That's the same for depositional environment. It will automatically tell you in your delimitation of space in the Scandinavia, I found that these are the depositional environments. Which of these do you want to keep? And it will talk to you. I will show you later in the practical part how the workflow is talking to you. And you will then select based on your preferences, for example, which depositional environments do you want? And they will be kept and then move to the next section. Okay, which we can do. And the next section is very big. It's chronologies. Or re-estimation of the ages. So basically, the workflow will automatically recreate all age-depth models for you for the compilation of your creating using the bchron, which is Bayesian probabilistic methodology to make sure that all the age-depth models are created in the same way, so they're comparable. It will also automatically add calibration curves, which are needed for if you do age-depth modeling with radiocarbon, you need to calibrate the radiocarbon dates. And they will automatically assign them to specific cores by geography. And if needed, if you have very young, for example, samples, if you also add this specialized post-bomb calibration curve, you don't have to worry about it too much. This is all done automatically within the workflow. What will also create, it will use these chronology control tables, and these are specific data within Neotoma for your specific course, for example, where it's stored, for example, the information about the radiocarbon dates, etc. So it is up to you, the user. You can select these types of these chronology control points, and it could be radiocarbon, biostratigraphic, tephra, etc. Right? And again, the workflow will kind of talk to you and said, hey, look, I found out that in your compilation, these specific chronology control point types are present. Which of these do you want to keep? Which do you believe they're valid? And it's up to you as a researcher to kind of select, oh, I consider these types to be good enough, right? To have good enough errors for my research to be included and used to recreate the HF models. And in the end, you also may specify that how many of these dates or these points in these control tables are minimum number of them are needed to construct a, good agent models. So for example, you can say, oh, I'm okay if only 2 radiocarbon dates are present in the table to reconstruct these agent models, or you may want to be more, I would say, more heavy on your and want to be more precise result and say, oh, I only want to, let's say, at least 5 dates to be present to reconstruct the agent model. And this is again up to you. And much more, right? This was really big. This is a really huge section of the workflow, and there's lots of things happening. This took us a very long time to do. But if we go back to our example from the Scandinavia, now I applied some criteria. I only included radiocarbon dates. I selected that the agent models needs to be constructed from at least, I think, 5 control points from within each core, and I recreated all agent models for them. And those which were good enough in my criteria, there I kept in. And the gray ones, as you can see, these are drop out. So they do not fulfill the criteria of good agent modeling for me. Therefore, I'm discarding them from my compilation. Are there any questions at this stage? This was very technical, I know, but maybe there will be some general questions about agent modeling. If not, I will continue next. Okay. So we are back on this line. So we start with the configuration file to set up the workflow. Then we source the data, do spatial elimination, select ecology groups, select the position environments, went to chronologies, select what kind of chronology control points we want, recreate automatically all agent models. And the next stop on our journey in this workflow is harmonization. As I already mentioned in the introduction, this is done to reduce the taxonomic uncertainty of the data, right? So we are using something which is called harmonization table. And to simplify this, we will have 4 taxa, and one of them is, I don't know, there is slightly variation of the same name. It could be, I don't know, Pinus type, Pinus, right, etc. And we kind of know they should be the same just because the different people, different authors of the original data name them slightly differently or have maybe different reference collection, etc. We want to make sure that they are actually the same pollen type in the end, right? So we will create a table where we unify or harmonize all of them to the same value. But lucky for you, there's already, I would say, a big plethora of already published harmonization tables. For Latin America, it is not so easy, but we actually published a paper, which is approaches to pollen taxonomic harmonization in quaternary paleoecology. And in that paper, specifically in their supplementary, there is actually there are actually harmonization tables for each continent, which you can use as kind of a starting point. Right? You still need to make sure that the harmonization table is good enough for you. And here I will really stress to work with expert palynologists to make sure that your harmonization tables are, make sense, are good, and build it for your paper. Because you can imagine, for example, again, I will go example from Europe. If you are interested in changes in humans, then it's really crucial for you to make sure that the cerealia is kept separately out of Poaceae and not harmonized into Poaceae. But for some other different projects, you may be interested. It doesn't matter. So you will kind of try to reduce it to, let's say, family level, etc., and you'll just harmonize everything to Poaceae, etc. Any questions regarding the harmonizations? I will give you a minute. There's something in the chat. I will open here. Okay. Then we will continue.
Sorry. I have a question. See,
Yeah.
I couldn't. I don't understand how do you do their harmonization? How do you reduce that? I was thinking something like when you work with bacteria and there is this O2 with some kind of, you decide the percentage of similarity to some DNA, but I don't know how you can do that here with this pollen that also maybe are not existing anymore. I don't know. I'm not an expert. But can you explain that again, please?
Yeah, sure. So how this works is that the workflow will provide a list of all pollen types currently presented in your compilation. And this will be the first column. And then in the second column, you will kind of give a name to what there should be harmonized to. So if you have, for example, same variant of the same pollen types, and there may be a typo in one of them, right? There will be two different similar names, and you should know, oh, they're actually the same. Someone just did a typo. So in the next column, you will put them to harmonize to the same name together. It's very simple. It's just two columns basically in a table. The one is the original name, and second is the name to be harmonized into. And this will be then used and harmonized through the whole dataset compilation.
Okay, I understand. Yes, yeah. Thank you.
I will try to quickly. I will try to open one actually and show this on a screen, hopefully. So here is the example. So there is the taxon name. Here you can see it's ABS and ABS undifferentiated. So we know that they both just need to be ABS, right? Or Acer competitor type is both Acer. So this is the original name, the first column, the second column kind of just have the name to be harmonized into, right?
I have a question. Hello?
Yeah, go for it.
The harmonization is for all taxonomic levels? I mean, for example, can you harmonize families or genera or something like that? Or is only at species level?
Again, it is up to you, right? The workflow will provide a list of all taxa, of all pollen types which are presented in your compilation. And it's up to you you need to create the harmonization, right? You need to set, oh, we will harmonize them to family. So you need to fill in the families they are in. It's not automatic. You actually need to provide this information. So if you will harmonize it to family, you can do it. If you want to harmonize to species, mostly you will be not able to because pollen types are mostly not going into species level. But you can try, right? But it's up to you.
Okay, thanks.
So to add to that, in terms of harmonization, especially when you work with data that hasn't gone through, for example, Neotoma, you know, people have different ways of naming the same pollen type because they add a point or a slash or whatever. So if you would not kind of clean that, then it seems that you might have more pollen types than actually exist. So one of the first thing is just to make sure that there are no kind of basically different names for the same thing. That often happens when you have data sets from a lot of different people, right? The other thing is that even when you work with Neotoma, for example, when these kind of harmonizations are often done with experts that basically say, you know what, working in this area, there are certain pollen types that are probably not, that you cannot actually identify at the level that people propose that they do. So we're just going to kind of group this together because we cannot actually differentiate very well. So it's often done with experts that have gone very deep into the sense of differentiating between pollen types. The initial step is just to kind of clean and get to a level that is further down useful. So with this step, I would definitely work with regional experts and just go through the whole list that later on will go through your analysis. So.
Thanks. Diana, I think, has a question.
Yes, thank you. I just wanted to know how sensitive is your workflow to spaces on capitals versus non-capitals letters, just to have that noted.
So, so it is 100%, right? So the name, so there is actually some, okay, to be honest, there is some internal processing which is happening to make sure that this is clean before, for example, the spaces or the types. So that's why you see in the harmonization table I showed, it's already, everything was turned into snake style. I will show it again. Can you see this, hopefully?
Yeah.
Can you see? Yeah, okay. So here you can see that spaces are taken out. So everything is translated into, I would say, computer readable code, right? So everything, there are no special, for example, special symbols, right? There are no spaces, etc. So everything is and everything is into the lowercase automatically.
Okay.
So this is happening for you to make sure we're kind of get rid of these potential issues. But and then and the next column is how you want to, in the end, to be present the taxa to be named.
Thank you.
Does this answer your question?
Yes, it does. Thank you.
Okay. Can we move on, or more questions? Feel free to ask if you want. We'll see. Okay, let's move on, and you can ask later if you want. That's fine. So we went through the input, the sourcing, the chronologies, the harmonization. Now, the final step in our journey is the filtering. And so here you will select samples and whole data set based on your criteria to increase the quality of your data. But it's completely optional. You don't have to do any filtering. But if you work with a lot of data and you want to do a big synthesis, let's say continental one, I highly recommend. Right? So you can filter by, for example, pollen sample. You can say, oh, we will only include specific samples if they have at least 50 pollen grains, right? Because there are some which maybe have just 10. And you say, oh, this is actually not good representation sample. So we will just filter them out. We will just throw them out. You can also, of course, filter by age limits that you're only interested in, let's say, as I mentioned, for last 8,000 years, right? So you just say, oh, there needs to be at least between 0 and 8,000. You can also filter by something which is called age extrapolation, meaning how far is the sample from the last age model control point, which means how far you're extrapolating your ages. And this is kind of a measure to limit your uncertainty from the extrapolation, right? Because then the extrapolated values will be very high errors of the ages, will be very high uncertainty. You are not very sure how actually old the sample is. And of course, you can filter out by the whole number of samples by saying, oh, we just want to, for example, only cores which in the end, after all this filtering, have at least, let's say, 5 samples, right? Because to do some reasonable temporal variation. And much more, right? Again, there are quite a lot of things which you can do. These are just examples. If you go back to the example of the Scandinavia, I applied some criteria. I think 150 pollen time, as I mentioned, last 8,000 years ago. Extrapolation not more than 3,000 years, and I think each needs to have 5 samples. And this is, again, the colored one are those which are kept within, which I said, oh, these are good enough for me. So your argument may be, and you can see here, which we go from sourcing to chronology to filtering, that we started with almost 300 records in Scandinavia, and then we slowly filter out, and we end up with 100 only. And you may say, oh, you're throwing out so much data. But I think our argument is that we are really increasing the quality of the data for our specific research question, because we are pretty sure that the answers, the research question we're going to answer with these will be more solid because of the quality of the data which is going to go into the analysis. Any question regarding the filtering or this data quality? Give you a minute. You can think about it. If not, we will now focus what is actually the output of what you will. Oh no, what have I done? Okay, of the output of the data, right? What you will gonna get out. Oh no, why is this happening? This is so annoying. So you will get a bunch of things automatically output, right? The first thing, it will automatically create a pollen diagram for each core at the end of this process. For each basically core or data set in your compilation, it will automatically create a pollen diagram in PDF. It will also save your agent models as individual objects, and you can look at them and share them. It will create some kind of graphical visualization of your data, which I will show later. Of course, there will be the dataset compilation itself in R, which you can then use directly to do your analysis in R. And one thing which I'm very, very proud of is the metadata citation. So I think I will go through them step by step, a little bit more in detail. So there's the dataset compilation in R, ready for analysis, right? You can immediately load it to RStudio and use it. There's the graphical visualization, which here, for example, for our example, it will plot it where they are on a map, so geographic location. It will also plot in the second figure is the temporal not resolution, but the temporal scale, right? So each line here is one individual data set. And you can see our focus was between now and 8,000 years ago. And this is where most of the cores are actually having the data in, right? So this is very good. This is what we want to see. And it will do some kind of sum of how many of them. I did this by WWF classification. You can choose whatever classification you want. These are the colors and also these kind of types of the data, right? These are biomes from WWF, but you can classify them as you want. And lastly, but not leastly, there is citation metadata, which I think I'm very, very happy that we did this. In the end, it will produce a table which will have the names and the contact information of all owners from Neotoma and references to the original data for your specific compilation. So meaning you can then either invite them to do a paper or at least acknowledge them in the, etc. Right? So I really believe in this acknowledgement of the work of other people. And it will also produce something which we call reproducible bundle, which is a zip file which contains the configuration file, all this, for example, harmonization table, or your specific things that you choose during your work, which you can share later with your paper to say, hey, if you want to recreate the same compilation as we did from Neotoma, these are the tools to do so. Okay, any questions regarding the outputs at this stage? Again, I will show you quite a lot of them, hopefully, in the second part, in the practical part.
Hi, Ondrej, I do have one question, if that's all right.
Hello, Quinn. Yeah.
Hi. So I was wondering if you were talking about like the species names and the like. I was wondering if you could mention any sort of potential dangers. Sometimes I'm worried when I'm using a software that does a certain amount for me. I'm worried that maybe it'll do something I don't expect it to. Maybe it'll rename something in a way that I don't expect, or so are there any like dangers of things that might slip through the cracks along the way?
100% they are, right? And it is software which is being developed. So one thing it is doing a lot of things for you. I agree for you, but at the same time, as you will see, it's kind of built in a modular script where you can. It's just a bunch of R scripts in the end, and you can go in and edit lots of these scripts if you want. You can add steps. If you're a skilled programmer, you can add individual steps to do some different things if you want, etc. But if you are really beginner, you can just basically run the scripts one by one, and it will do lots of these things for you. Regarding the names, as I said, you are creating the harmonization table, and so it is up to you to make the names. If, for example, but there are ways, hopefully, which you should help you to track some issues. So, for example, when you do the harmonization, and it will find out that there is a taxa in the compilation which is not present in your harmonization table, it will tell you, hey, by the way, this amount of pollen grains were left out during the harmonization because I don't know what to harmonize it into, right? So I try to build it in a way that it is safe, right, as much as possible. But it depends on your skill of the, I would say, on the coding side. But you will see in the practical things, I really try to make it, or we try to make it in a way that the workflow is kind of talking to you, right? And if it's not clear, as always, I will show you there is a ways contact any of us to make sure that this is happening, or what to improve that this is not clear from the outputs, and we can go in and change these in the end.
Yeah. Okay, that's great. So there's like a defensive programming level already built in, which is great.
Yeah, we tried.
Thanks.
Okay, so I'll summarize it hopefully. So the FossilPol is created this workflow which handles all these steps for you, as we see, and flag places where the user need to specify their choices. I will show you how this works in practice. And we publish this in a paper, which is the title is A Guide to Process a guide to the processing and standardization of global paleoecological data for large-scale synthesis using fossil pollen in global ecology and biogeography already 2 years ago. And I'm very happy that I think a month ago we received that we actually got award for the top cited article in GAP for that year. So people are citing the paper and using the software, which I'm very happy. I think it's a good paper in general, where we spend lots of time about, what you should consider when you're creating this, multi-core compilations, and potentially use FossilPol. But if you don't, you can code it yourself, right? But for some kind of guidelines. Oh, you need to remember that these are actually the things which are affecting, right? Like the depositional environments. Lots of people forget about that different cores come from the depositional environment, which may have actually impact on the, I would say, temporal pattern and trends we are observing in the data, etc. Okay, and as I discussed, there's a lot of information, right? This is a big project. It's a complicated project, technically complicated. So to make this as easy for the users as possible, because we want you, the potential user, to use it, right? I want as many people as possible to use it. We created a website, and I will now show you the website. You can either just Google FossilPol, or there's bit.ly/FossilPol. I will try to open this now. No, I didn't want to click that. I want to click open a new site. So I will open a new tab. So this is our website. I will go through them right now and show you where everything is. One important thing, which I really, really want to thank, especially Nick Hoffman, because there is now Spanish version of the whole website. So for those who, English is not their primary language, as for me, and they're struggling, we try to do Spanish version. There may be mistakes in the translation. If you spot any, please let me know, and I will fix them. Right, but you can switch between English and Spanish as you want. I will be now showing the website on English because I don't understand Spanish, but all the sections are the same, just in Spanish. So there is I would say the index file, kind of the home page, which is telling you what the workflow is, what the FossilPol project is, who are the authors, and what different sections are. So there is the link to the paper, which I just mentioned in GAP. There is link to where you get the workflow. I'll show it later. Where you get the package, which I'll show you later. There's the websites, right? There is the Scandinavia example. So the code to run the case study for the Scandinavia, which I showed you. You can go there and click it. There's issue tracker. If you find any specific issue or something is not working, go to issue tracker and let me know. I maybe open this, all of them. So for example, this issue tracker. I'll just open it here. So it is GitHub issues. So if you have any issue of something that's working, you will click new issue, and you will either request a new feature or report the bug, and you will just post it, and I will immediately get notification that something is not working and it should be fixed. There are lots of things. There are some feature requests already. Most of them are by me for the kind of future things which I would like to do. But I definitely, this is open for anyone, and you can just make a new issue if you want. There is kind of what is present currently on the website, and there is what are the versions of the individual moving parts, I would say the software happening here. So I will start with some general information, right? So there is, again, the general information about what the thing is, right? It is our studio project. So it is a bunch of R scripts which are doing all these individual steps. There is the figure which we just saw, which is kind of the individual steps which are happening. There's the first step, which I think is the most crucial thing, how to obtain the workflow, how you're actually getting the software, right? So you either use it as GitHub template, which I'm not sure if you. So if you are, I need to get. Can I hide this? This is so annoying. So if you click this link, this is the workflow on GitHub. If for those who are familiar with GitHub, you can just use this template if you have GitHub account, and it will work at any other template. If you don't, there is this second option for those which are not so familiar, which is fine. You will click this. Again, I will open it in the tab, and it will get you to this releases. And here, this is the newest version. And basically, click on the source code, and it will download a zip file for you. It's taking some time for me because I'm streaming, but it will download the whole thing, and I can show it now, hopefully. Here. Oh, this is my download. Don't look at this. This is all secret, super secret stuff. But here is the zip file, and I can look into this, and there is actually the R project, which are I hope quite a few are familiar, and I will show it again later how this has been done. Okay, so this is how to get the workflow, right? So, and the workflow, we can kind of skip to that, is just a bunch of scripts which are organized in this way that there's folder called R, and in it there are folders like data processing and main analysis, etc. And in them there are additional folders, and each of them, and there are individual R scripts. So there is a lot of scripts, but you don't worry. You can either run the whole workflow with the one button, with just running one script, and it will call all the other scripts, which is, I think, described here. No, or you can run it script by script individually for those more advanced, I would say, programmers. So I will get to the setup later. I just want to first show you what is there. The next big part is step by step, which is really detailed description of all individual, I would say, steps you need to take from the data sourcing to the position environment. And there's description of individual scripts which are there, what they are doing, and how you can alter them, right? So we really spent quite a lot of time on this documentation. So it's going to be quite a detailed documentation about how to prepare chronology control tables, for example. It will tell you what this script is actually doing for you to understand. If you're not so familiar coder, you can read this about calibration curves, what they mean, why they're, how this is all happening, etc. And the next step is get in touch. So if you want to contact us, there is a GitHub discussions which you can fill in. There is also, if something doesn't work, it doesn't work, right? Sometimes that may be a chance. If you find out something is not working, you can again send us this issue report, as I showed you previously, or you can open a discussion. I will open this. There is GitHub discussions now, which you can start a new discussion, etc., to kind of say, hey, I'm going to talk about this. This is kind of forum for you. Can we add this? That's a very common thing, as we heard. Like, can we add this proxy, for example? Yes, if you want, please start a new feature request. I'm not saying this will happen, but we can try. But you can kind of see, I think there is a future update thing where I will show you potentially what I'm doing, right? So this is, I guess this will be in the future. This is currently in consideration, all the things which I'm doing. In the next version, I haven't planned much because the development been kind of stopped by other projects. But as you as a user, you should be able to know what is coming in the future for FossilPol as the software. And if you want to contribute, right? If you want to, we will be very happy. If you're a coder and you are interested in this, or if you just want to get better, this is open source, so we're more than happy to actually invite anyone who wants to just help us make this better. And there's ways how to do this. And last but not least, there are resources. There's resources relevant to this one, like the paper which we mentioned, but also previous presentation which I showed about this, some posters, and the logo. And I think I will also add this recording of this seminar will be also there in the future. Any questions regarding the websites right now before we jump into, I would say, the RStudio and show you how to set up everything? Yeah, okay, all good. Diana is just saying good, I guess. Okay. Okay, so I just want to mention, right, that we have developed this paper, and right now we've been using it also, right? Because this is, I think, cool, very cool tool. I hope so. I hope you will also find it cool. And we published already some really cool, I think, interesting paper using this FossilPol, right? So obviously this is me and Suzette. 2021, we published this big paper in Science, which basically used the similar thing, where we gathered over 1,000 FossilPol records from all over the world and estimated rate of change, and it was published in Science, and we are very happy. Also, we published kind of synthesis on a Asian level, where we actually use both Neotoma and also private sources. And right now, we also have a really cool preprint about looking into climate and human effects on vegetation during Holocene, which is available. And there are other papers, like there's African kind of synthesis by David Early, which are in preparation, and there are more, which I just thought I will highlight some of the papers which already have been using. These are mostly done by us, but if anyone else wants to, use this for their paper, I will be very happy. So this presentation, which I mentioned, is publicly available. I hope Alejandra will send the link to anyone and you can walk through it as you win and click all the links and everything on your own pace later. And that's kind of everything from this first section. And I thought we will now jump to our studio, and I will show you how actually to set up the thing, how this thing works, etc., and how it works in practice. Are there any questions before we do that? Yes, Diana?
Yes, have you considered to link some of your data with the BN database, or to run some taxonomic check with tax size, or not? Okay.
No. So again, we are only using the data from Neotoma or private
Okay.
Sources if you want, but basically no, it's just FossilPol, right? Like you mean for the harmonization, or with like
No,
for
just
which part?
for unifying the workflow. Like sometimes I just download data and then I have to go to the BN first to tax size to double check taxonomy and names and things like that, and then to BN to link climate information. So I was just wondering if that's something that you have foreseen for the future. But
So this is a nice segue, which is not part of this, but I have another project which is called VegVault, where I did exactly that. And I can, is it fine if I insert my advert? I hope it is fine. But
please.
I'll just, so if you do VegVault, so there's a VegVault. Oh, this is great insight. So which is a database linking paleo and neoecological data, functional traits, and climate. And as you just said, right, it's basically, I will show you. So it takes Neotoma and BN and S-plots and Tri and Chelsea and VOSIS, and it all puts them into a unified database, and everything is kind of, we use some automatic, as you said, taxonomic clarification to make sure everything is on the same taxonomic classified, to GBIF backbone, actually, etc. So, but this is completely different project, but which is using the FossilPol as one path to this database. But this is completely separate project, so I will, which I can give talk some later, some other time.
Thank you. I will, with this, I will track it down. Thank you.
Any other questions before we jump? Suzette, do you want to comment on something? Are you happy? Is it all fine?
Yeah, I guess the part here relevant to mention to everyone is that a FossilPol is really built in a way that it should kind of lead you intuitive through the different steps. But the ideal scene is that you give it a try even for like a test region, even if it covers like 5 records or something. So you get a sense of it, and a lot of the background information is provided stepwise. So on the website it's quite detailed, but if you do that in combination with the way that we have also the paper kind of described step by step, then it becomes a little bit more realistic, I think. Yes, that's what I just want to say.
Okay, then what I will do, I will jump to our studio. So you know, we downloaded, as you see, we use the releases, we downloaded the project. I just opened our studio project. This is the project. This is actually the Scandinavian example, right? I will kind of use that as an example because it's small enough, so it will not take that long. And one thing which you need to do, and I will maybe jump back to the, you need to set it up, right? So you need to first set it up to make sure it is working for your project. I will jump back to the website where here in the section how to obtain workflow, then it's set up. So there's actually in detail, you need to make sure that you have R and R Studio, right? This is crucial to be running because it is our project. Then there is a script which is called init project, which means initiate. So it will basically make sure that your all installed packages and everything will be working, hopefully, right? This is just, and it will take just some time to install the packages. And then this is the crucial point. There is the config file, as I mentioned previously, which is stored in 00 config file, R script. I will open it. And here you as a user are specifying the choices you want to do, right? We use this user flag, I will show you, which is the thing where you need to specify mostly. So let's go to R Studio. And here I actually have the open, the init. Here you can see the script called init, which is the initiation. You don't have to worry that much about it. It will just do the thing for you. You can just run it one by one. But most importantly, there is this config file. And again, what I try to do in R is, I'll make this a little bit bigger, is that it's always, each script has a header. So you will know that this is a FossilPol project. What is the script name? Who coded this? And some kind of description of what the script is doing. So here is the configuration script, and you need to really pay attention to something which is called user, right? So if we scroll back here, for example, we see there is a first thing which is a user flagged, kind of. It's commented as a user. And here we want to say where lots of the data should be stored, right? Because if you're doing a big project, like let's say the whole world, if you want to do the global project, there will be lots of data, and you can specify where exactly in your computer will be stored. If you don't change this, it will automatically be within this RStudio project. So it will download her in this folder we show you. And let's look here. So there are some other variables which you need to want to choose. So for example, set geographical boundaries. These have the user, right? So longitude minimal, longitude maximal. So this is the rectangle as we saw about the geographical boundaries. I want only data which is between 5 and 30 longitude and between 55 to 73 latitude, right? So this is roughly Scandinavia. You can also select it by altitude. You say, oh, I'm only interested in like high altitude cores from like mountain regions. You can also set it there. We have it. If you leave it NA, it will just take all of them. And there are lots of settings, right? So there is this some kind of HDIP model settings, which is the minimum number of chronology control points. So the chronology need to have at least X control points to run. We set it to 5, etc. Right? So this is the config file, which is configuration of lots of these things within the repo, and you just need to go through it, look for these user flags, and change it if you want. If something is not clear to you, for example, this min, I would recommend to go to a website. Again, I will go there, and if you go to step by step, you can just look for this specific phrase and it will find it, right? We can try. Yeah. Boop. Hopefully it will take us there. Maybe. No. So this one, it will not take us there. Yes, it will take us there. So it is in this section somewhere. I will try to do it like this. So I can look up for this specific phrase and it will explain where this is used and what is it doing, right? So we always have kind of the documentation for if something is not clear. We try to make it as clear as possible already here in the script, in the configuration script. But you can always look it up on the website, which is more details, I would say. And there is lots of description, including some kind of graphical, etc. Okay, so this is the setting everything up. So we kind of set it up. We choose our configurations. And now we can run it, right? So again, I will show this on the website. If we go to general information, you can what the script is doing, what the workflow is doing. It uses Cascade, Here we describe it. Oh, sorry. It uses cascade of R scripts, meaning you can just run the master run and it will do everything. Or you can run the individual scripts. So here is the example. Here are some scripts, download Neotoma, extract samples. You can run them one by one, or you can run this 1001 and it will run all of them by itself. Right? So the simplest one is the master. If you run just the script, it will run the whole workflow. It will do everything for you. Or you can do it one by one as you want by individual scripts and change them. So in practice, here, hopefully, if you're familiar with RStudio, here at the bottom, I have the folder structure. Here is the R, where all the R scripts are living, right? Here is the initial script, as I didn't mention. There's the configuration script, and there's the first folder, which is data processing. And I can just run this master run. If I would just run this script, it will do everything. It will do all the individual parts for me, and it will do everything. Or I can go step by step, so I can get into first section, which is the sourcing, Neotoma source. And again, I can run this script, which will run everything inside, or I can go one by one. I can use this script, this 0.1, download Neotoma, right? So, and it's, you know, to Quinn's question, these are just scripts. You can, if you want, they're mostly using functions from my own package, but you can add any other code which you want to do with them, or check something, or etc. If you're a skilled coder, you can do this. But there are just a bunch of scripts to do the thing. So we can even show this. So for example, when in this download Neotoma, and I can, as you know, you can just highlight anything and just press run, right? And it will run it. Or, and I recommend this a little bit more, you can press this source button and it will run the whole script, right? So I will press it. And now this is the example of the workflow is talking to you, kind of, right? So it will say, I start to download Neotoma files. Now it will take some time because it's actually checking with the server for the Neotoma. I haven't, hopefully it will be fine with me streaming, but let's give it a moment. If not, we can do, yes. So, and you see it says, oh, I'm extracting coordinates for all records, and it will take, you know, it's 50%. So there is even like things you can watch how quickly it is doing, and it will do the steps and hopefully tell you what it's currently doing. So it will start with Neotoma. It says that it's get the list of Neotoma sites successfully obtained. It find 298 records within our geographical boundary. And it find out that 9 records can be downloaded, right? So since the time I did it, I don't know, a few years ago, there are 9 new records in this area, and it will download them. It will only download those which needs to be downloaded automatically. If you once downloaded them, you don't need to do it again. So it's kind of smart. And you see there was a progress bar, and now it says all selected records were downloaded, and it saved the changes, right? So this script is now done, and I can continue to the next one, right? So I can extract samples again. If you want to read one by one, the description of each of the script are here at the website, step-by-step guide. And I will just click you there, right? So there is this download Neotoma. There was the script we just run, and it says exactly what it's doing there with some examples. Now, extract samples, there's the second script. It will tell you all the things which will be happening there. And I can just execute that, right? So it's extract samples from Neotoma JSON structure. Don't worry. And also extract the author information. Remember, which I mentioned that the workflow is kind of keeping track of all the information about the authors of the data. This should be due, and we can run it. And again, it will tell us, oh, I'm processing, oh, this is a lot of things happening. I'm processing Neotoma files. I'm loading Neotoma files. So it will load this Neotoma download from today. That's today's date because we have new update, which I downloaded just a few minutes ago. And we'll load it. Now it will extract Neotoma meta information. So it will say that all sites are prepared. The number of records is 298. Oh, sorry, it's going very fast. And that it extracted meta information. Now it's extracting samples. So both metadata and sample data have been extracted for all the records. It will start to download Neotoma author data. So it will extract information about 93 individual authors. And it will all add these authors to personal database. So there is actually a small database happening in the background, which is keeping track of all the datasets and authors there. And it says done. And save it, right? Saving the file. Okay, is this fine? Any questions until now? Like, this is just simple running the individual scripts going from the beginning at the end. Or you can just run one which will run them all automatically. But I like to do them step by step to make sure that I know what it's doing. Any questions until now? Maybe, hopefully some people, if you have any questions about R in general, right, I can try to answer. Not? I will go to the next one where I think this is deposition environments, and here there will be a different feature which we haven't seen before, where it will be talking to you again, and I will show you how. So I will source the script. So this is filtering based on deposition environments. So if I source this script, it will again. So it will tell me starting processing deposition environments. It will load the file from today. It will start preparing deposition environment section. It obtained all deposition environments from Neotoma, and it says it did not detect previous file. Continue to create new one. So it says have not found previous version of the file. Blah blah blah. Please open deposition environment section in a folder. You don't see this, but automatically it opened on my other screen. It opened a folder here, right, with a file in it, right? So let's continue reading. And so please open the file in the folder, in specific folder where is it stored, and select which deposition environments should be kept. Include weak equals true or false. Then proceed and rerun this whole script. So we will do what the workflow is doing. We'll go to this folder, which is it will automatically actually open the folder for you, and we can look. And there is a file which we can open. I'm trying to make it a little bit bigger. Right, so it has different. So this is how things are done in Neotoma, and it's tell us we should include true or false. By default, everything is false, and these are the depositional environments, right? So this first is the most important. So these are depositional environments which are found within our compilation. There is much more in Neotoma in general, but it will only show you those which they found in your compilation at this stage, and it's up to you to choose which depositional environments do you want to keep in your compilation. So for example, blanket bog, I want to keep in, so I will put true. Bog, I will also put true. Coastal, I don't want to put, so I will put false. I'm not interested in coastal things. I think this can mess things up. Drain lake, I want to include, etc. Right? So we will now go one by one. I will basically just take now everything, all these lacustrines. And terrestrial, I'm not interested in. Unknown, I'm also not interested in. If you maybe notice that there are some additional columns here. And this is a little bit confusing about Neotoma, but each deposition environment has their own classification, right? So for example, the blanket bog is within category called bog, which is within category called mire, which is within category of palustrine. And there is no higher than palustrine, so I will repeat palustrine, palustrine, palustrine. Right, so for example, then you can see raised bog is within bog, which is within mire, which is within palustrine. So this will kind of give you a insight about where roughly they are, what they are, right? What these deposition environments are. And I now it will ask me to choose to include true or false. So I include it. I save the file, close the file. I will go back to R, and it says rerun the whole script. That's exactly what we're gonna do. We'll go back to the script. It will take source. And again, let's see what it's doing up. And it's a lot of things happening. So it will list all the deposition environments from Neotoma. And it says there has been 16 selected deposition environments. It will list those which we set as true, which we want to include. And based on that, it prepared 283. So we dropped previously, I think it was 897. So we dropped like 10 different records because they didn't fulfill our criteria of deposition environments. And it will again save the file, right? Is this clear? We call this stop check table because the workflow will stop. You as a user need to check something or make a decision and then continue. Is there any questions about that?
Is there any way to know which records, I mean, or having a list of which records are you dropping out, like save somehow to easily ID them?
Everything is being saved, but
Okay, then I have to cross
so
them. Okay.
so what you can exactly, right? So
Yeah.
what you can do, if you know a little bit of R coding, right? So these are Neotoma meta sample deposition environments filtered. This is the one which is creating the filtering those one. So we can it's an R object, right? So we can just take a look on that, and this will be the meta information about them, right? Data set ID, handle, site ID, site, longitude, latitude, right? So you can compare this one with the one which was done before, which is this meta samples, and you need to manually compare them, right? So there is not easy way. It's not keeping track of which of these are filtered out, but because they're individual R objects, you can do the comparison if you want, if you know how to do this. But as I said, these are just a bunch of scripts doing the individual, I would say, simple things. So we will load the data, test the presence of the data, filter the data. This is the function to filter the data, and then save the outputs. Any questions? If not, I will maybe a little bit jump from the R scripts to the important part, right? Which is the outputs. So there is, not surprisingly, there is a folder called outputs. And here there are different things. So there is meta and references, which there is this data assembly meta, which hopefully we can view. Now, not like this. Okay, then I will open it. Here, sorry, I will just open it in my browser. So outputs, meta information, or dataset assembly meta. So automatically at the end, it will create a table with all the meta information about the files in the end, all the process data, right? So these are, in the end, we have this, I don't know, 100 something, 114 records. These are all their IDs. So the handle, site ID, site name, longitude, latitude, all the information you want to keep about the general overview of these 100 we have at the end, which is cool, I think. We also have this author table where, no, sorry. No, hello. Where you will have the names of the people and which dataset they are which they uploaded, right? So this is a link for you to make sure that these are all the authors of the dataset compilation at the end for you, right? So you can contact them. They have emails if they have emails in Neotoma, which I think is really good. Then in the outputs, we also see the data itself, of course. There's this assembly, which is kind of our RDS, which normally you will load in R. And then there are figures. There are lots of figures, right? We can look at pollen diagrams. And this will be the ID. They are named by the ID. And we can take a look. Oh, no. And this is a pollen diagram for that specific dataset ID in that time you want. And it has several pages because that's actually to be able to print them. So they're all there. We also have in the figures, we have a chronology, which is actually the HDEP model chronology. You can visually, if you know a little bit of HDEP modelings, you can then visually observe how the HDEP model for that specific dataset looks like, if it's good or not. It's also present there. Oh no, these are some supplementary. Okay. So I think that's kind of, for me, that's kind of it. We're hour 20 in. So I think that if there is more space for questions, I'm more than happy to show or answer or something was not clear, or maybe some question in Spanish if you're shy in English. Suzette might try to answer. I hope it was understandable for myself. Alejandra, do you want to comment question?
So I think like if there are no more questions, then we can just say that I would like just to say, so we're recording this meeting and we're gonna make it available on the Neotoma YouTube channel, so everybody can follow it and then maybe review it again, because I know there's a lot of information here, but it's very useful. And then also we're gonna send a follow-up email with also additional information. And this is like the last event that we will have of this like series of talks and workshops that we organize. But so please stay tuned, because we're planning to have another like series of talks and workshops starting next semester, probably September. So thank you very much for to everyone for being here and asking questions and being patient too. And also especially thank you very much to Ondrej and Suzette for hosting this workshop. I think it's been great and it's been very informative. I'm sure that people will find it very useful.
Yeah, I just want to mention also, maybe I understand it's overwhelming and there's lots of things happening, but if you decided, and I hope you decided to use this tool, and something is not working or something is not clear, please reach out to me, right? The user experience that say, oh, this was confusing, I wasn't able to set it up, the information was just, please tell me, or Suzette or anyone, to make sure that we make this better, right? Because in the end, we're making this for you, for other people, right? And we really want people to be able to use it and to make it clear and super. So any feedback from you to us is super valuable.
Yeah, I also wanted to say one last thing. Like a couple of years ago, Ondrej, as I said, also hosted another workshop that we organized with the African pollen database, and there was one based on a different R package. So I'm also gonna share that information, because I think like you can also find it very interesting. So if you also have questions, just please reach out. We'll be very happy to hear from you, and also have comments if you think if there's something that needs to be improved, or if you are interested in also maybe hosting an event next semester, that will be super cool too. So we're always looking for people that can help us to build like this kind of workshop series, so we can share more knowledge in general, like on paleoecology and bioclimatology. Okay, well, thank you very much everybody for being here. Hope you have the good rest of your day, if you're still early in the morning, or not, like good evening or good night. We'll see you around.
And thank you very much, Alejandra, for organizing everything and for inviting us. It was great.